Good afternoon. I'm introducing Dr. Richard S. Bedlack Jr., Director of the Duke University ALS Clinic, presenting the Outer Limits, Updates and New Directions. Directly after Dr. Bedlack's Q&A, you will have a break from 3 to 3:15. Please be sure to visit our expo booths and learning lab during that time. When you need to ask Dr. Bedlack a question, please make sure you put it in the chat, and it's all yours, Dr. Bedlack.
Thank you so much. Great to see everyone. And I'm sorry that I can't be with you live in person, hopefully next year. But at least I get to present to you on a very special day today. I don't know how many of you know this, but it's National Batman Day, which is why I'm dressed like this. We just got back from several different comic book stores here in town. So I'm excited to bring you some updates and some totally new directions with regard to my research work, which, as you know, is a little bit different. And so today I've entitled this The Outer Limits in homage to an old TV show, which I believe was from the 1960s. And I'm going to go ahead and play the intro to that show as the intro to my talk. All right, so what are we going to talk about today? I'm going to bring you some updates on some of the things I've talked about before: the ALS Clinical Research Learning Institute, the ALS Untangled Program, my ALS reversals research, my pilot trial of Clenbuterol, and my Neurophoresis work. And then I'm going to take you to some new directions, some things that I've just started working on over the past year. One is expanding access, and the other is something I call high throughput drug screening in people with ALS. And so I do have some disclosures. I have active research support from the ALS Association, the Healy Center, Medicine Nova, and Orion. And I consult for several companies, as you can see here, mostly running their data safety monitoring boards or helping them to design more patient-centric protocols. So let me tell you a little bit about how I got interested in ALS and got set down this path to work on all these unusual projects. I saw my first person living with ALS around 1998. And you can tell that this picture is from that era because I had a Jerry Seinfeld hairstyle, and also because it was the only time in my life when I wore blue blazers. I was a resident at Duke and I didn't want to make any waves, so I dressed very conservatively. And I'll never forget that person's story or the collection of findings I saw in their physical exam, because it struck me that it was the most fascinating and most terrible presentation I'd ever seen up to that point in my life. And I couldn't wait to tell my attending physician about this and see how we were going to help this person. And he kind of breezed in and said, "This is called ALS. We don't know why it happens, and there's really nothing we can do about it. Just go home and get your affairs in order." And that didn't sit right with me. And I think that was the seed that was planted in my brain to build the Duke ALS Clinic, which I started in 2001. And it's grown to be what I believe is one of the largest and most comprehensive ALS clinics in the world. And also to be able to incorporate research, all different kinds of research in that clinic, to help people to feel like they're contributing to the greater good and also to have some hope. But amongst the many surprises early in my career in the Duke ALS Clinic, I noticed that not that many people were actually getting into the research studies I offered. And that led me to a couple of surveys, which ultimately showed me that was just the tip of the iceberg as far as the problems that we have with ALS research. The big problem is that patients, at least back then, were just not very engaged in our research. We were asking them to be subjects in our study, but we were not actually getting them involved in helping us ask the right questions or design protocols that were inclusive and were doable and gave us an answer that was meaningful. And so I decided I would have three big missions in life. First, I wanted to increase the number of options, including experimental options for people with ALS. Second, I wanted to improve patient awareness, understanding, and involvement in research. And finally, I wanted to design a different kind of study than most people were offering, one that was more inclusive and more attractive. And so I looked around to see if any other fields were noticing similar problems with patient engagement, and sure enough, many of them were. And one of the most interesting approaches to optimizing engagement, I thought, came from the folks who worked in Parkinson's disease research. They had created something called a Parkinson's Clinical Research Learning Institute. It was a set of classes that would train patients and family members to better understand the research process and then meet with them regularly to help them influence and improve it. And through this program, they were finding ways to engage patients and family members at every step of the research process, from the study concept to the funding, to the protocol, to the implementation, to the interpretation of results, to the dissemination of information, and even to the presentation for regulators like the FDA. And all this was leading to more studies, better awareness, studies with more relevant questions, with better designs, easier consent forms, and better enrollment. And so I decided to work with my colleagues in the Northeast ALS Consortium to create ALS Clinical Research Learning Institutes, which I abbreviated ALS Curlies, CRLIs. And so far, we've had 19 of these, and you can see where and when they took place. But through these 19 clinical research learning institutes, we've trained more than 400 patients and families to be what we call research ambassadors. And we meet with them regularly every month through something which is called the PEACE Committee, Patient Education and Advocacy Committee. These are just basically video conferences like the one that we're doing, where we ask all of our research ambassadors to come on, and then we invite stakeholders from all across the research space to present projects that they want some patient insight into. And so these stakeholders could be researchers like myself, they could be patient advocacy groups like ALSA, MDA, IMALS. They could be sponsors. We've had several companies like MT Pharma present protocols that they wanted some help from patients with. And the graduates of the program, the research ambassadors, have just blown me away as far as all the things that they're doing. They have helped to raise hundreds of millions of dollars for new research studies. For example, two of the guys who were in one of our first clinical research learning institutes went on to start the Ice Bucket Challenge. They have lobbied in front of Congress and helped pass new laws and budgets that facilitate more research. For example, their efforts helped us get and continue to fund the National ALS Registry and the Department of Defense's ALS Research Program. They are spreading awareness. They are answering questions live at support groups and fundraisers and online on many different websites and chat rooms. They're helping us design better ways to find trials, and they're working with researchers and sponsors to design better trials that will be more inclusive and attractive. And finally, they're working with regulators like the FDA to help them understand patient perspectives. And if you saw the news this week, the efforts of the research ambassadors and many other patient advocates from across different organizations resulted in something that I have never seen in my career as a physician. The FDA actually changed its mind about a product that it was presented several months ago called AMX0035. Initially, the FDA said they would not be able to approve it without another trial. And as you know, doing another large phase 3 trial will take several years. And there was a huge blowback from the patient advocacy community, first directly to the FDA and then to Congress. And as a result, we learned this week that now the FDA is saying, we'll take another look and we'll see if we can find some creative way to approve this and get it into the hands of people with ALS soon. Another thing that surprised me early on were what I call alternative and off-label treatments. I had no idea how much interest in these types of products there would be amongst patients and families living with ALS, nor how many products were out there. There are hundreds of thousands of these products currently being advertised on the internet. And unfortunately, information about these products is not always complete or even accurate. And this could lead to an overestimation of the potential benefits of buying some of these products, and even an underestimation of some of the potential risks of these products. And so I started this program in 2009 called ALS Untangled, which would systematically review these products, ultimately helping people with ALS to make more informed decisions about them. And there's 3 parts to this: inputs, where do we get our ideas? Reviews, how do we actually know what to say about these products? And then outputs, how do we get the information back to people that might be interested? And so inputs come from patients. And right now they come mainly through email. And once somebody emails an idea to us, it gets listed on our website under a section which is called Future Reviews. And by the way, our website is www.alsuntangled.org. And if you go to this page of our website, you will see that we have over 400 of these AOTs that patients have asked us to review. That's probably more of these things than I can do in the remainder of my lifetime. And so we've got to find some way to prioritize these. And we figured out that we would do it in 2 ways. First and foremost, by votes. So anybody can vote for the AOTs that they're most interested in hearing more about, simply by clicking the vote button next to the name of the product. The other way that we prioritize is by something called a multiplier. So we multiply the votes by either 0, 1, or 2, based upon the amount of useful disclosable information that we can find. For example, if a product has a lot of votes, but there's absolutely no useful disclosable information, well, we can't write about it until something becomes available for us to write about. So we have to multiply the votes by 0 until such time as the information becomes available. Most of the time, the multiplier is 1 because there's a little something out there for just about everything on this list. The multiplier becomes 2 if there's at least 1 published clinical trial or case series in any disease on that AOT, because that's where we learn the most about a product. In terms of how we do the reviews, well, we had to invent a way to do it because there is no similar program out there, still isn't, for any other disease. Even though I think all disease states have the same issue with patients wanting to try AOTs and not being able to get good information about them, nobody has taken this bull by the horns and tried to do anything about it. And so we invented standard operating procedures that guide everything we do, from how we gather information to how we write the draft and crowdsource it across our team, which now consists of more than 120 clinicians and scientists from across 11 different countries, to how we ultimately publish our drafts. And all of these SOPs that we invented revolve around something called the ALS Untangled Table of Evidence. So basically, we're looking at 5 different categories for each one of these reviews. The first one is mechanistic plausibility. What is this product supposed to do? And does that make any sense as far as biology, and in particular, the biology of ALS? The second is preclinical. Has this product ever been given to any kind of ALS model, a cell model, or a disease model like an animal model? The third is case reports. What do we know about people who tried this? What are they saying happened to them? Do we have any records we can get to corroborate what they're saying? The fourth is trials. Was this ever in any ALS trials? And if so, what happened? And then finally is risks. What exactly can happen to somebody who takes this as far as harms? And within each of these 5 categories, we assign a letter grade, which ranges from A to F. Just like when you're in school, A is what you want, F is not what you want. And you get a better and better grade based upon having better information. So for example, if we're focused on the case reports category, if all I can find is one person on Facebook who says they took this product, say they have ALS, and say something good happened to them, but I can't prove that this person is real, they're not responding to my attempts to find them, I don't have any medical records, then you get a D grade. But if I can find that person and get their records, and their records tell me they really did have ALS, and something objective really did improve in association with this product, then you get a C. And if there's more than one person like that, you get a B. And if there's a lot of people like that who are published in a journal, that gets you an A. So that's how that works. So once somebody writes a draft, it gets circulated across our team. The entire team has 2 weeks to send in their comments and suggested edits, and then I incorporate those. And then I send the crowdsourced draft to a journal, send it to the main journal in our field, which is called ALS and Frontotemporal Degeneration. And it goes through one last peer review in that journal, and then I have to respond to that. And then finally, it gets published. And we have an agreement with the journal that all of our reviews are published through something called free open access. And that means nobody ever has to pay to read one of our reviews. And we also publish our reviews on the completed reviews section of our website. So if you go to that section and you click on the name of what you want to know more about, it will open up the published review. If you don't want to read the entire review, you can just look and see what we gave it for grades across those 5 different table of evidence categories. We also know that nowadays, many people like to listen to their information. And so we've partnered with a group called Create, the Create Consortium, to do podcasts where one of our research ambassadors comes up with some questions about each of these published reviews and then interviews me. And that recorded interview is available just by clicking on that microphone icon next to the AOT that you're interested in. And we've learned that there's interest in this program in other countries that are not necessarily fluent English speakers. And so our reviews are now starting to be translated into other languages. This past year, thanks to feedback from our research ambassadors, we did a complete overhaul of our website. So if you haven't been on there in a while, I would encourage you to check it out. The update included a much more searchable website, a way to alphabetize the reviews, updates of older reviews. So every week I try to update 1 or 2 of these, and you can tell which ones have been updated because they have a little asterisk next to them. So this program occupies a lot of my time. There's hardly a night or a weekend that I'm not working on this program in some way, either reading papers about an AOT or trying to find a person who's out there saying that they're using this and improving, or trying to get myself invited to a clinic that's selling an AOT. It's fair to say, is it really worth all this? Well, I can at least tell you that people are reading our articles. So our journal keeps track of its most downloaded articles of all time. And we have 8 of the 10 most downloaded articles of all time in the history of the journal. Some of our individual reviews have more than 40,000 downloads. 40,000 downloads for one paper. That's probably more downloads than all the rest of the 140 or so papers that I've published, some of which are in pretty prestigious journals like Neuron and Science. So clearly there's interest. Collectively, the series has over 200,000 downloads. The podcasts, even though they just started about a year ago, have more than 12,000 downloads. And I'm really proud of the fact that my peers, other neurologists from across the world, recently gave us an award. It was a competition for which organization was using technology in the best way to improve patient care. And we won that award. It's called the Neurology Brainstorm Competition. The other thing I can tell you is that I've learned a ton from leading this program for the past 12 years. I've learned things about the people selling these products, that many of them have a good heart, just that they're not really all that knowledgeable about science. I've learned that many of these products are actually quite interesting and worthy of further study. But without a doubt, the most interesting thing I learned is that some people with ALS can actually recover from it. I found my first ALS reversal about 10 years ago now, in the midst of an ALS Untangled review on energy healing. And with the help of social media, I found a lot more since then. As of today, I found a total of 53 people whose medical records I have. I've been through their medical records. I agree with their doctor's diagnosis of ALS. The records suggest to me that these people did progress to where they were disabled from the disease. And then they suggest to me that they recovered a significant amount, in some cases all, of their lost motor function. So these are pretty dramatic recoveries. I mean, some of these people were in wheelchairs and now they walk and sometimes even run. Some of these people had lost all the use of their arms and hands, and now they're back to being able to, do their own activities of daily living, and in some cases even work on a farm and milk cows and build snowmen with their grandkids. Some of these people couldn't speak or swallow, and now they do that normally. So I don't know why these cases happened, but what I do believe is that there's something really important here, and that I think we really need to study these folks, because if we could figure this out, we might be able to make it happen more often. And I've got a couple of different types of research underway to try to understand these cases. The first category is called STAR, Studies of ALS Reversals. I'm trying to find as many of these as I can, and I do continue to get records. It's a dull week that somebody does not send me a set of records. Unfortunately, most of the time, I'm not able to confirm either the diagnosis or the improvement. I'm trying to get all these people into the same database for the first time so we can compare their demographics, their disease characteristics, their comorbidities, the products and medications they took, to people with more typically progressive ALS. I'm trying to get biological samples on as many of these people as I can so that I can look at genetics, proteins, RNA, microbiome, histology. And I'm trying to ask these folks as many questions as I can think of about unusual things they might have been exposed to that might explain these cases. And so far we've got this one paper that we've published on the first, I believe it was 39 ALS reversals. And these people do look a little different than typically progressive ALS. They're a little younger, a little bit more likely to be male, a little bit more likely to have limb onset disease. They progress a little faster before they start to improve. And maybe most interestingly, they're more likely to have taken certain products. And this table over here is the products that are more likely to be associated with ALS reversals in databases compared to people who are in there with more typically progressive ALS. And the odds ratio tells you the odds of that product being associated with an ALS reversal versus more typically progressive ALS. And in research, we typically get excited about odds ratios of 2 or 3. Well, the odds of a person with an ALS reversal being exposed to curcumin are 348. So that's a very interesting product, which we'll get back to in a little while. I'm happy to say that since that first case, there's a lot less skepticism about these cases than there used to be. I have now a lot of collaborators around the world that are interested in these cases, interested in helping me study them. So I've got people in Duke Neuroradiology helping me to go over the imaging in these patients. I got the CREATE Consortium helping me to look at the genetics. I got ALS TDI helping me to look at the RNA and proteins in these patients. I got the Duke Microbiome Center helping me to look at the composition of the gut microbiome in these patients and comparing it to patients with more typically progressive ALS. I got the experts at the National ALS Registry helping me to analyze exposures. And I've got the NIH Neurobiobank agreeing to help me look at the autopsy tissue. Hopefully many years from now, when these folks die of natural causes, we can look at their brains and spinal cords and see, is there anything different in there compared to people with more typically progressive ALS who are in the same biobank. So that's one type of research. The other type of research explores this hypothesis. Is it possible that these cases could be due to the treatments that these people took? Now, that may seem far-fetched, but there are many other examples of patients discovering drug effects that researchers had not thought of. This is called serendipitous drug discovery. The most famous example of it is Viagra. Pfizer was studying Viagra for heart disease. It would have been scrapped had it not been for the fact that men who were in the heart disease study came back at the end and said, I don't know if it's doing anything for my heart, but it's doing something else I really like, and I'd like to get a prescription for some more of this if I could. And the researchers said, I had no idea that it might be a treatment for erectile dysfunction. They changed gears, studied it for that, and lo and behold, it became the first ever drug for that problem. It was $1 billion blockbuster drug that was discovered by patients, not scientists. Nuedexta was discovered in the same way. We were studying it to try to slow ALS progression. It didn't work, but patients came forward and said, this really seems to be helping that laughing and crying problem that we had, which you called pseudobulbar affect. Company switched gears, studied it for that. It works beautifully. It's the only FDA-approved drug for pseudobulbar affect. So to work through this idea, I've created a program called ROAR, Replication of ALS Reversals. So this is a series of small pilot trials of the AOTs that are associated with these dramatic ALS reversals. Now, because most of these products are generally regarded as safe by the FDA, many of them are sold in grocery stores and on Amazon. And because this program is looking for the largest signal ever in an ALS trial, we're not looking for a 20 to 30% slowing. We're looking for recovery of lost function. These ROAR trials can be very different than what you typically see coming from a pharmaceutical company. These trials can have very wide inclusion criteria. Almost anyone can participate in a ROAR trial. These trials do not need to have a placebo. I can use historical controls. I don't need a lot of in-person visits. I can teach people to measure things, and then I can check in with them virtually by phone or by video to make sure that they're measuring what we want them to measure and entering it in a database. And the database that we're using is a widely available database that's available free to everyone with ALS. It's called Patients Like Me. It's a really nice database that you can go into and create a profile. And you can put the products that you're taking, and you can even put in the measurements that either you or your clinic doctor makes so that you yourself can determine in that database whether there's any products that seem to be correlating with you doing better. But by using this as a database in a trial, it does one other thing that I really like. It makes the results available in real time. Since this is publicly accessible, anybody can go into there and check and see, patients using a product that's in a ROAR trial, what are they saying? What is it doing? So you don't have to wait years for the results of one of these. And then one other thing that we're doing different is we're publishing our protocols on the website, www.alsreversals.com. And that's because, again, we know that there's many people around the world that want to experiment. They can't get into a trial. And so they're kind of on their own trying to determine which AOT to take. So if we give them a roadmap that they can take to their own doctor, at least maybe they'll take something that is a little bit more plausible and a little bit safer than some of the other options that are out there. So we have completed a ROAR trial. We studied a product called Lunasin, a peptide that came from soybeans and appeared to have an interesting mechanism and was associated with a very dramatic reversal of bulbar ALS. And there was some good news and some bad news about this result. The bad news is this product did not appear to be as safe as we thought it would be. Many of the patients in our trial had side effects. GI side effects were very common, including cases of constipation so severe that people had to be hospitalized. This didn't seem to affect any biomarkers that we could measure that suggested that it would do the things we thought it needed to do to help a person with ALS. And it didn't seem to be helping anyone in our study. We found no evidence that it was slowing, stopping, or reversing ALS in any of the 50 people that we had on this product for a year. But the good news is this very unusual, controversial design worked amazingly well. It was the fastest enrolling trial in the history of ALS. Most ALS trials enroll 1 to 2 patients per site per month. We enrolled more than 9 patients per site per month. The people who participated in this study were very diverse, much more typical of a clinic population than the people who participate in most studies. That means if we ever do get a result from one of these Aurora trials, it's much more likely to be applicable to everyone with ALS than the typical result from a very restrictive clinical trial. In spite of the fact that we took people who were further along in their disease, in some cases 20 years in some cases with feeding tubes or tracheostomy or ventilators, they stuck with us all the way through a 1-year trial. They continued to enter data. So those patients that were alive at the end of the study, 84% of them were still entering data just the way they were supposed to. And we found that they could and would measure exactly what we asked them and trained them to measure on a monthly basis. And then finally, by publishing our protocol, we learned that we empowered hundreds of people around the world outside of the study to try something that we at least thought was reasonable with their own doctors. And so, of course, we're going to do more Aurora trials. And the next Aurora trial is going to be, or is, a study of curcumin. What is curcumin? Well, it's a natural product that comes from plants. It's bright orange. That's the molecule. It's from the chemical class polyphenols. This is naturally found in spices like turmeric and curry powder. We did an ALS Untangled review of curcumin a few years ago, and it was, I have to say, one of the most challenging reviews we ever did. For the opposite reason that most reviews are challenging. Most ALS Untangled reviews are difficult because there's not much out there for information. This one had information overload. There were more than 12,000 scientific papers published in journals about curcumin. And I have to say that they fell into 2 groups. Some of the papers said it was the greatest thing since sliced bread, and other papers said it was complete garbage. And I think the reason for the discrepancy is that there isn't just one curcumin. There's a lot of different ways to package this molecule. And so you can package this in what are called nanoparticles, or you can add a bioenhancer like black pepper. All of these things change the pharmacokinetics of curcumin. They alter its ability to get absorbed. And so this got pretty good grades for an ALS Untangled review. Mechanisms grade C, because it modulates neuroinflammation. It's an antioxidant. It impairs protein aggregation. It even alters the fecal microbiome, at least in animals, which I think is very exciting. Preclinical grade was a C because this was given to a cell model of ALS and it seemed to have benefits. Cases grade B because it's associated with 9 different validated cases of ALS reversal. Trials grade C because there have been 2 clinical trials of different forms of curcumin in other countries. Now, there were a lot of problems with these trials, methodological problems, but they were considered to be positive trials. And finally, risk grade a B, because when taken orally, at least, curcumin, all of its different forms appear to be very safe across hundreds of different human trials in different conditions. And so we had to think about, what's the best way to dose this? How much is it going to cost? Well, there's no head-to-head trials of all the different forms of curcumin that are out there. But after a lot of reading and discussion with experts, I've come to believe that this one particular product called Curcumin from a company called Integrative Therapeutics at a dose of 1 capsule twice daily is probably the way to go. So there are pharmacokinetic studies of this product that show it's getting absorbed into the blood and probably into the brain. There are animal studies of this exact product showing that when this is given orally, it protects mouse brains from oxidative stress. A very similar formulation was associated with better survival, fewer tracheostomies in a trial. In another 18-month trial, this exact formulation and dose seemed to improve cognitive impairment in 21 people, while 19 people in the same study on placebo got worse. And maybe best of all, this product is pretty cheap. You can get it for about $50 a month on Amazon. So we now have the Aurora curcumin trial underway. Again, it's a widely inclusive trial. We want 50 people with ALS. We want 50 healthy controls. We have 36 people with ALS and healthy controls enrolled so far. Every person with ALS who's in this study is getting the actual product. We're teaching them to measure their ALS functional rating scale, their weight, their perceived effectiveness, and their side effects. And we're comparing these measurements over 6 months to matched historical controls from the Patients Like Me database. We are also very interested in the microbiome. And so that is part of this trial. We're looking at the microbiome at baseline and how it changes over time with Curcumin. And we are going to be asking the healthy controls in this study to give us microbiome samples so we can see how a person with ALS has a different microbiome than a healthy control from the same household. This trial has no in-person visits, all virtual visits done by phone or by video. All data is being entered on Patients Like Me. All biomarkers are being collected through the mail, so you don't have to go anywhere. The results of this study are available in real time on Patients Like Me. In case you're thinking of doing this protocol with your own doctor, the protocol is published on the website alsreversals.com. So hopefully next year I'll be able to tell you the results of that study. But I want to shift gears for a minute and tell you about another product. Now, my ideas for testing things in ALS come from different sources. Some come from patients, some come from ALS Untangled, some come from ALS Reversals. Others come from the literature. Sometimes I find things published in papers that for some reason were never followed up on that look to be incredibly promising. And Clenbuterol is one of those things. What is Clenbuterol? Well, it's a beta agonist that is used to treat asthma in many countries around the world. It is not FDA approved for any indication, but it is often used illegally by athletes. So if you're an American sports fan, you may have heard about your favorite baseball player or cyclist or boxer getting suspended for testing positive for what they call Clen. Now, why would an athlete want to take an asthma drug? Well, it turns out that there are beta adrenergic receptors on many cells throughout the body, including in the brain and spinal cord, and even in the muscle. And this drug appears to activate those receptors in muscle, stimulating protein synthesis, making muscles bigger and stronger. Well, that's cheating in sports, but it's exactly what we would want to happen in a person with ALS. I am not the first person to think of using this product in people with ALS. 15 years ago, somebody gave this to the mouse model of ALS and showed that it improved motor function and survival. 15 years ago, a neurologist in Italy gave this at a dose, a very low dose of 20 micrograms 3 times a day to 16 patients from his clinic. And he published this paper, and I thought the results were pretty extraordinary. So this table right here shows different outcome measures. Time 0 is at enrollment, time 1 is at 3 months, and time 2 is at 6 months. And I want to focus on the upper limb and lower limb myometry. These are strength measurements in the arms and legs. And look at what happens here. On average, for this group of 16 patients, the scores are higher 6 months in than they were at baseline. And that turns out to be statistically and clinically significant. We can also look at the breathing capacity. On average, the FVC was higher at the 6-month time point than it was at enrollment. And that turns out to be statistically and clinically significant. There are not many trials that I'm aware of in the history of ALS where people on average actually seem to be better 6 months in than they were when they started. We usually get excited when we slow progression, 30% or so, but to actually see an improvement is highly unusual. I don't yet know why this was never followed up on. I reached out to the people that did this study, and their response was they just kind of got interested in other things, just kind of lost interest in this. But I decided to study it with a generous donation from a family here. And again, I did a very widely inclusive study, 25 people with ALS, everybody got the drug. And I compared how they did during 6 months of treatment to how they were doing before treatment. The slope of decline in their ALS functional rating scale and their forced vital capacity. I also looked at muscle strength testing and safety labs. And we decided to use a higher dose. And the reason is, first of all, most studies do use higher doses than the ones that the Italians used across many different conditions, including studies by a co-investigator at Duke. There's a neurologist at Duke who've used this drug in kids with muscular dystrophy. And he felt like kids could tolerate much higher doses and that we might be able to get much better benefits out of that. We tried to make this as easy for people to participate in as we could. There's just 4 in-person visits and 4 telephone visits. And we finished the study now about 7 months ago. So what happened? Well, here's who enrolled. So pretty typical of an ALS population, age 60, a little more males than females, except for the fact that the people in this study had their disease significantly longer than people in most studies. And it's easy to see why. You know, we offer a lot of different kinds of trials in our clinic, and people who are newly diagnosed are getting into the trials that are offered by pharma. Typically, they want people who are in the first 2 to 3 years of their disease. Most of my patients can't get into those trials, and so they're hungry for other trials that don't have disease duration criteria. So they jumped on this study. Most of the people were on riluzole. So let's first look at safety and tolerability. So the good news is it was safe. We didn't see any worrisome safety lab changes. We didn't see any serious adverse events that were related to Clenbuterol. But it wasn't as tolerable as we hoped, at least at the doses that we chose. So 13 people dropped out of the study before 6 months was up because of side effects. That's a little more than half of the patients that we enrolled. And the main side effects were cramps and tremors and insomnia and stiffness. And we know that this whole class of drugs is associated with those kinds of side effects. We just didn't know they would be this severe in people with ALS. One other person dropped out of the study because of the COVID pandemic. He didn't want to come in for any in-person safety visits. So that was the bad news. The good news, of the 11 people who finished the study, every single one had slower progression on their ALS FRSR score during treatment compared to how they were doing before they started treatment. One person had no progression for their entire 6 months on Clenbuterol, and 4 people actually had an improvement in their ALS FRSR score on Clenbuterol. They were previously getting worse, and they actually improved over 6 months on this drug. And so if we do what's called a per protocol analysis, including only those 11 people who finished the trial, on average, this is a 70% slowing in progression on Clenbuterol compared to how people were progressing before, which is obviously statistically and clinically significant. Now, some people don't accept those kind of analyses. They say it's not fair to only study people that completed the study. You should try to study as many people as you can through something called an intention to treat. So we couldn't figure out how to study people unless they had at least 2 data points during Clenbuterol treatment. Now, why? Well, because we're calculating slopes. And so we use the 2 data points to calculate a slope of progression. And so when looking at the ALS FRSR, we found 20 people that had at least 2 data points on Clenbuterol. There, 15 out of 20 had a slower ALS FRSR progression on treatment compared to how they were doing before. Overall, about a 37% slowing on Clenbuterol compared to how people were doing before, which again is statistically and clinically significant. If we did intention to treat analysis of breathing measurements, forced vital capacity, we found 16 people that we could include. And you can see the amazing results in that graph right there. This showed a 90% slowing in ALS FRSR progression on treatment compared to how people were doing before treatment, which again is obviously statistically and clinically very significant. So I'm very excited by these data. Again, this is not the first time somebody showed this, but I think it supports the animal study. I think it supports the small case series. I have some patients now who are importing Clenbuterol from other countries through a group called the Social Med Work. I'm hoping to raise money to get this into a much larger, more traditional study. Personally, I would love to see Clenbuterol become part of the Healy platform trial. The next project I want to update you on is Neurophoresis. So one of the great things about working at Duke is there's so many smart, creative people. And I've got a colleague, a neurosurgeon, who's invented this really cool device that he calls Neurophoresis. It's a way to filter the spinal fluid. So it's basically a catheter that he inserts in the bottom of your back, and it takes spinal fluid out, it runs it through a filter, and then it puts back the filtered spinal fluid into the same person. Now, why did he invent this? He invented this for people with something called subarachnoid hemorrhage, which is bleeding in the spinal fluid. So what does this have to do with ALS? Well, it turns out that the spinal fluid of people with ALS is toxic. If I take one of my patient's spinal fluid and I put it on astrocytes or microglia in a culture dish, it changes them. As far as astrocytes, it makes them secrete pro-inflammatory chemicals, and it impairs their ability to regulate glutamate. As far as microglia, it fires them up. It makes them very destructive and pro-inflammatory. And maybe most amazing of all, if I inject that same spinal fluid into a mouse or a rat, it actually causes an ALS-like disease in that animal. So let me repeat that because I think it's astounding. If I take spinal fluid from one of my patients with ALS and I inject it into the spinal fluid of a mouse or a rat, that animal gets an ALS-like disease. Amazing. What's in the spinal fluid that does that? Nobody knows for sure, but we are suspicious that it may be small inflammatory chemicals called cytokines and chitins. We know that these chemicals are different in the spinal fluid of people with ALS than they are in healthy people. And we know that if we take these chemicals by themselves and put them on astrocytes or microglia, it can change those cells. What we don't know is whether this filter can get rid of these chemicals. These are much smaller than blood cells. And so I told you, I think last year, that we've got this proof of concept study where we're getting spinal fluid from people with ALS, and we're running it through our filter to see what comes out. And then we're injecting the filtered spinal fluid into our animals to see, did we remove the toxicity of the spinal fluid? And I want to just show you one of our early experiments because I think it's pretty exciting. So this is the experimental setup. This is the side view of a mouse's brain. His eyeballs would be up here. This is the back of his head, and this is where they're injecting the spinal fluid of a person with ALS. And this is the assay, basically putting a mouse into a plexiglass box, and we're watching him with a camera, watching what he does. And so take a normal, healthy mouse that we didn't do anything to. The mouse is very curious. You will see that the mouse walks all around the box very frequently. Now, if I take a mouse and I inject it with the unfiltered spinal fluid from a person with ALS, you can see that the mouse appears to remain curious. His head's moving all over the place, but he's not moving around the box much, and that's because his legs are weak. Now let's take a different mouse and inject it with filtered spinal fluid. And it looks like we restore normal function. So the filtered spinal fluid no longer appears to be making these mice sick, which is incredibly exciting. So we need to replicate this. We need to try to isolate exactly what it is that we're getting out, and then we need to go to the FDA to see if they will let us do a clinical trial of this device in people with ALS next year. So these are just the tracings of what these different mice did. So in the last part of my talk, let me just address a couple of exciting new things that I've gotten involved in over the past year. The first is expanding access. I told you in the beginning that just about everyone I've ever met with ALS wants to try experimental treatments, but most of them cannot get into formal trials. And so there's only a few options for trying experimental products outside of a trial. Expanded access programs, which are FDA-approved programs. They've been around since the beginning of the HIV era. There's very few of these, unfortunately. The right to try pathway, which was passed across almost every state and then nationally signed into law by President Trump, and it was hoped that that would improve the paucity of expanded access. It hasn't, and that's because that law actually addressed the wrong problem. Expanded access was never really about the FDA. That's not where the bottleneck is. The barriers to expanded access are at the level of the physician, who doesn't understand the program very well and also is challenged by the resources that they have to devote to it, and by companies that own these products and are reluctant to give them away outside of their clinical trials. And so how are we circumnavigating some of these barriers? Well, thanks to a generous grant from IMALS and a matching award from Biohaven, we have one of the largest expanded access programs in the history of ALS underway, running in parallel with the Healy platform trial. So this is initially going to be 35 people who get access to this at 3 centers, and eventually we hope to expand it to 100. We're also using some of this grant to try to create new materials that will educate physicians who want to provide their patients with future expanded access programs. And that's going to include an annual lecture on how to do this, as well as materials like templates and training and advocacy materials that'll be shared with the whole community. So hopefully we can knock down some of the barriers to doing expanded access. There also are a couple of bills in Congress you may have heard about, 8662 and 4867, which propose, among other things, the creation of a federal fund that could be used by investigators who want to offer expanded access programs and study their outcomes. And I'm keeping my fingers crossed that all the amazing patient advocacy that we're seeing gets these across the finish line, because I think there'll be a lot more expanded access if these get passed. Now, last but not least, I want to tell you about an idea that I've had in my mind for a long time, but couldn't think of how to do this. It's called high throughput drug screening in people. So what is high throughput drug screening? Well, it's a proven way to accelerate drug development. And it basically works like this. Let's say that I wanted to try to find an existing FDA approved drug that could kill COVID-19. What I would do is I would create an assay where there was some COVID-19 in thousands of individual tubes like this, and I would ask a computer to squirt every FDA approved drug into one of those wells and tell me which drugs we already have that kill COVID-19. It's a very fast and accepted way to find drugs that hit preclinical targets. And we've used this to get ideas for trials. Like, for example, this is how we got an idea to try ceftriaxone. It's also being used in preclinical models. So people use this in worm models and fly models and mouse models of ALS. They screen hundreds of drugs in these models to figure out which ones prolong survival the best. But we don't do this in people. Now, why don't we do it in people? Well, because each time you set up a formal experiment in a person, it takes months and sometimes years. You've got to create a protocol. You've got to get people under contract. You've got to get funding. You've got to get FDA approval. You've got to get institutional approval. But here's the thing. Experiments in thousands and thousands of people with ALS are happening every day all across the world. It's just that we don't have a way to pay attention to them. There's no easy way to track the results. So it occurred to me that what we need is a tool to track the results of all of these N of 1 experiments that are going on spontaneously around the world. And so I've already been using historical controls to predict what's going to happen to patients in my Aurora trials. What if we developed a tool that would put this in the hands of individual patients all over the world, and they could work with their own doctors to say, well, I'm going to try coconut oil, or I'm going to try cannabis. And I want to know how I'm expected to do over the next 6 months versus how I actually do. And so we're calling this the PET, Patient Empowerment Tool. And it's about to launch. What you'll be able to do is register on patients like me, put in some information about your ALS so far, and it will generate, using historical controls from the database, what your progression curve is expected to look like. You can then go in on the products that you choose with your own doctor and put in the outcome measures that you're actually getting over the next few months and see, are you progressing right along this curve, in which case it's not obvious that stuff is working? Are you doing significantly better, in which case there might be something here? And we are going, as the scientific community is going to be able to get alerts about regimens, could be one product, could be 50 that somebody's on, that are associated with doing significantly better than a patient was predicted to do. And this will now be a way for us to get ideas to bring right into clinical trials. So I'm very excited about this. So just to kind of summarize, I've shown you that we've got ALS Clinical Research Learning Institutes empowering people with ALS to greater engagement, and amazing things are happening, including making the FDA reconsider drug approvals like AMX 0035. I've shown you that we've got this program called ALS Untangled that's helping patients and families make more informed decisions about experimental products called alternative and off-label treatments. I've shown you that we've got 53 ALS reversals and how we're studying them through the STAR and ROAR program. I've shown you this pilot trial of Clenbuterol that I did that I'm very excited about and hope to be able to replicate it in a larger, more traditional design, like the Healy platform trial in the near future. I've shown you some very preliminary but exciting proof of concept results from our Neurophoresis work. I've shown you how several of us are trying to expand access to products that are in development. And finally, I've shown you this new tool that I think is going to provide individual patients and the community with high throughput drug screening in people with ALS. I couldn't have done all this without a lot of help. So first and foremost, let me thank all the people with ALS that are listening. You are literally the reason why I get out of bed every day, and I'm so excited and I work so hard. I want to find more and better options for you. And I want to especially thank all the people with ALS who've participated in the projects that I showed you today. Got to thank all of our sponsors, especially the ALS Association for funding ALS Untangled, all of our collaborators, the ALS Untangled reviewers, patients like me, Integrative Therapeutics, Minitronics, Drs. Coble and Ladd, the medical students Dan Harrison and Jesse Crail, who've done so much of the ALS reversals work. And finally, the Duke ALS care team. You've helped me build a foundation of great patient care onto which all of these ideas are built. And so I will leave you today with the final ending to the Outer Limits TV show. Okay, let me see if I can get back to the other screen now. Take your questions. All right. I see myself for the first time today. Whoa. So I am in the chat and ready for questions. Okay, I got some questions here that just popped up. Let me roll through this. Well, thank you. Thank you. Some nice comments here. One person asked, ordering through social network requires a prescription, but isn't it illegal for doctors to prescribe it? I do not believe it's illegal for doctors to prescribe it. If you go onto the FDA's website, they do have a pathway called the Personal Importation Policy, PIP, that allows a person with a difficult, currently incurable, progressive, disabling, life-shortening disease such as ALS to import products from another country for their own personal use. So that is the most important part of this. You cannot import products and sell them out of your car. That would be illegal. But my understanding of this is that you can import products from other countries for your own personal use. And it is not an easy thing to do, which is why I think the social med work is such a great option. It does require a prescription. It requires a letter of support from the attending physician. That letter has to say, I agree with this. I'm going to monitor my patient carefully. And the patient has agreed that it's for their own use. They're not going to sell it. The social med work then finds a doctor in another country who prescribes the medicine, sends it in. Social med work helps get it through customs and gets it right to your house. Now, it's not cheap to do this, but it's not as expensive as some of the other things that I've heard about my patients trying. All right, let me scroll through here again. Exactly where does ALS come from? That is probably the most important question of all. So about 10% of all ALS is genetic. Some might argue 12%, but somewhere around 10%. That means we can find a gene that is causing the disease. So in the rest, there may be genetic susceptibility, but there are also almost certainly environmental influences. And what's complicated is that this equation is probably not exactly the same in everyone. In other words, there probably are certain susceptibility factors that match up with certain environmental exposures. If you have one but not the other, you don't get ALS. That's complicated to figure out. As far as environmental exposures, well, we know that being a military veteran makes you about twice as likely to get ALS as not being a veteran. We know that physical fitness is probably a risk factor for ALS. Two other things that I think are very interesting. One is a toxin in the environment that comes from many different places, probably most commonly from blue-green algae. It's called BMAA, beta-methylaminoalanine. Some pretty good evidence that that might cause some cases of sporadic ALS. And we might even know about a way to block the effects of that toxin by using an amino acid called L-serine. There's a big trial underway out of Dartmouth right now to figure out if high-dose L-serine actually makes people with ALS progress more slowly or stops it or reverses it. One other potential trigger that I find fascinating is there might be a retrovirus, which is called human endogenous retrovirus K, that triggers the disease in some people. Now, we all have this retrovirus in our DNA, but in only a fraction of us does it appear to get reactivated. And that seems to be much more common in people with ALS than it is in healthy people. And so the NIH has a trial underway of an antiretroviral cocktail, the same one that we use to treat HIV. It's taking people that have this retrovirus in their blood and ALS, treating them with this cocktail to see what happens to the retrovirus and what happens to the progression of the disease. And I think we're going to have some results from that in early 2022. So I think that's really exciting. Are there any studies currently using CRISPR? I don't know of any studies that are currently using CRISPR in ALS. And part of it is, CRISPR is a gene editing technology. There are some studies underway that are attacking the types of genetic ALS we understand the best, which are SOD1 ALS and C9orf72 ALS. They're trying to knock down the protein products of those genes, which seems to be a really smart strategy. And I have great hopes for those studies, especially for the SOD1 studies, which I think we'll see results from here in the next few months. I think those would be great for the subset of people who have ALS from those causes. I don't know if those things will have any effect on people with ALS who don't have those genetic causes. But to be useful for CRISPR, I think we would have to figure out what gene do we want to target and how do we want to rearrange it to make it beneficial to people with ALS. And there may be some other genes that are more amenable to that technology than SOD1 and C9orf72. Is there a way to get into one of your studies? Well, we do have a waiting list for the Curcumin study. It's pretty long. We have, unfortunately, about 200 names, and we need 16 more people. So realistically, I don't think that most of the people on that list are going to get into that study. But again, the protocol is online. You can follow the exact same protocol with your own doctor, and the product is something that you can buy off Amazon. Certainly, we hope to have more of these kinds of studies. They're very resource intensive, but we hope to be able to always offer one of these kinds of studies in parallel with pharma-sponsored studies, because again, most people cannot get into a typical pharma-sponsored study. So we want to be able to have options that are open to everyone. Have I ever used dalfambatine? I have not. Can we talk about autophagy? Yeah. So autophagy is a process by which cells get rid of waste products. So like, for example, every day cells have proteins they don't need anymore. They need to get rid of these old broken proteins. There are organelles like mitochondria that are not functioning properly, and cells need to get rid of those. And that process seems to be gummed up in most people with ALS. The question is that something high up in the cascade of things that are going wrong, or is it really way far down near the end? No one knows the answer to that. If it's way up high, then promoting autophagy might be a really good strategy for treating ALS. But if it's way down at the bottom, when there's already a dozen different things going wrong, any one of which can kill motor neurons, then it might not make any difference. What's out there as far as autophagy promoters? Well, there's a few things out there, including products that are what I would classify as AOTs. So, for example, melatonin is something that we recently wrote an ALS Untangled review on. And that's another one of those things many of my patients used it for insomnia for years. And I never knew that it could have any connection to treating ALS. But at pretty high doses, it's a promoter of autophagy. And there's some really good mechanistic and preclinical data suggesting that this is something that we should put into a clinical trial. The question is going to be the dosing. Most people for sleep use a dose of like 2 to 3 milligrams. To treat a neurodegenerative disease, you might need much more, like 30 to 50 milligrams a day. But I definitely think it's worth exploring, things that affect autophagy, especially if they're simple. Let's see. We got one person that wants to try Clenbuterol. How can they get it and at what dosage? Well, again, it's not something you can just go to a pharmacy and get. So you would have to get your doctor to write a prescription, to write a letter of support. And then you would have the option of either finding a doctor in another country that would take those things and send the medication from that country into you through the FDA's personal importation policy, or working with the social med work who would take care of all that for you for a fee. And so again, every patient that I know of in my clinic who's doing this has worked through the social med work. And if I was going to take this myself or do another study of it, I would probably go back to the dose that the Italians used. That would be a starting dose of 10 micrograms, not milligrams, micrograms, 3 times a day. And after a week, going to 20 micrograms 3 times a day, which is, again, much lower than what we studied in our trial. Question about the circulatory system. Yes, there is some evidence that the circulatory system could play a role. There's one of the genetic mutations that's very, very rare that has to do with circulation. I think it's possible that in some people, that's part of the problem, but it's a really difficult thing to study and a difficult thing to figure out how to treat. Is it true that in the Healy platform trial, patients are not permitted to take curcumin? So my understanding is that they're not permitted to take certain specific products that are in studies, like Curcumin. But as far as using, turmeric or curry powder that have curcumin in it, I believe you'd be allowed to use those kinds of things. Any comments on the disease process, upper versus lower motor neurons? So classical ALS involves both upper and lower motor neurons. But there's certainly a spectrum that we see. There are some people who appear in life to have only their lower motor neurons affected. We call that progressive muscular atrophy. Interestingly, most trials exclude that form of the disease. I don't agree with that, because if you look at some of the studies that have been published on progressive muscular atrophy, they have the same epidemiology, they have the same disease course. At autopsy, most patients with progressive muscular atrophy have evidence of upper motor neuron involvement. So I believe that progressive muscular atrophy is a form of ALS, and I wish more studies would include that in trials. The opposite side of the spectrum is pure upper motor neuron disease. That's called primary lateral sclerosis. So most people would say that if you have pure upper motor neuron disease, you really need to wait at least 5 years before you can be sure if that's all it's ever going to be, or if it's going to turn into what's called upper motor neuron predominant ALS. In my experience, most people who start off looking like they have PLS actually do develop some very subtle but real evidence that their lower motor neurons are affected. And therefore, what they have is upper motor neuron predominant ALS. In general, that's a really good form of ALS to have if you had to have it, because it progresses much more slowly than more typical forms of ALS or progressive muscular atrophy. And again, to me, the real difficult thing is that these extremes of phenotype are often excluded from clinical trials, which is frustrating. Where can we find your studies? Well, some of these things, as you heard today, are in progress. So you can just go on the database called PubMed. If you want to see our publications, we have about 140 or so publications. Just put in my name. You can go on the website, ALS Untangled, to read those published reviews. You can go on the website, ALS Reversals, to read the publications there or the protocols. And you can continue to watch, for these recorded presentations where I try to keep people up to date on the very latest things that we're working on, even if they haven't been published yet. Is AMX 0035 available yet? No, not that exact product. It's not available. The components of that product can be purchased. So the components include a supplement, an AOT called TUDCA, TUDCA, which is available on websites like Amazon, and a drug, a prescription drug called sodium phenylbutyrate, available only with a prescription. Now, by taking those individual products, are you getting the exact same thing that you're getting in AMX 0035? Probably not, but there's no way to know whether it makes a difference. And this is a question I get asked a lot. It's a great question. Like, for example, somebody might decide to take TUDCA, and they might say, well, which brand should I take? I wish I knew. It would be great if somebody took the time and paid the money to compare the purity of all the different brands of any supplement, but they almost never do. When those experiments are done, the results are quite shocking. Very often, supplements are completely different in terms of the concentration of what's in there and what else is in there, including sometimes things that you don't want to take, like heavy metals have been found in some supplements, even though they're not listed on the label. So we need a lot more of that kind of research. It's just that there's nobody out there that's paying for it. Tell us more about energy healing and ALS reversals. Well, so there's nothing more I can really say about ALS reversals than I said. I just got to wait for more data. As far as energy healing, I mean, it's something where there are certain people who claim that energy can come out of their hands and heal different diseases. Now, getting back to the way that we grade things, that would not get a very good mechanisms grade for an ALS Untangled review, because there has never been any study shown that proves that healing energy can come out of someone's hand. We know of no biological mechanism that can explain that. Does that mean it can't happen? No, it doesn't mean it, but I think it's very unlikely. Somebody's going to need to demonstrate that for me to be convinced that it's real. I believe there are ALS reversals that have been seen in association with energy healing, but an association is not the same as saying one thing caused the other. There's a lot of other people with ALS who've tried energy healing who've had no benefit from it. So it may be that these people who benefited, including the first ALS reversal I ever saw, maybe that was going to happen anyway, whether they did the energy healing or not. In any event, just to show you where my mind is, I tried to study energy healing. I tried to study the exact energy healer who treated that first patient, that first ALS reversal that I found, and he refused to be studied. His comment to me was, for those who believe, no proof is necessary, and for those who don't, no proof will ever be enough. So that's as I said before, I believe that healer had a good heart, but that is completely the opposite of what a scientist would say. Scientists believe that things have to be tested and demonstrated and replicated and proven. And that's how we can most certainly advance toward finding cures for ALS is through science. Do reversals start with common correlations? You know, it's a great question. I don't think so. I mean, I've talked to almost all these ALS reversals, or, you know, at least seen very detailed explanations of how it happened. And it's very different in different people. You know, in general, they start gradually. I don't recall any of the 53 ALS reversals that occurred suddenly. You know, from the time that somebody noticed they weren't progressing anymore to the time that they noticed they were starting to get function back is often many months. And this is why I tell people when they're starting a new product and their goal is to find an ALS reversal, it's probably not worth, you know, getting rid of the product in a day or a week or even a month if it's not reversing your ALS, because it's going to take time. I mean, if that occurs, it's probably going to take 5 or 6 months to notice that something in this disease is working. Let's see, we had some questions about other diseases, things that are working in other diseases, like, for example, Parkinson's disease. So absolutely, we've got to pay attention to what everybody's doing. I told you that's where I got the idea for the Clinical Research Learning Institutes. And Parkinson's disease is also very interested in the microbiome, like I am. In fact, I'd say it's even better demonstrated that the composition of the microbiome may play a role in how fast someone with Parkinson's progresses. And so I'm watching with great interest to see if anybody can isolate some specific pattern of organisms that seems to be correlated with better progression. That's what's missing right now. Like, I know a lot of my patients want to try to take a probiotic or do a fecal transplant. The problem is we don't know what the microbiome should look like to correlate with slower progression. Until we know that, we can't really know how to treat a person's microbiome. So hopefully next year, I will be able to tell you what a good and a bad microbiome look like, and then we can start talking about how we might get there. Let's see what else we got here. I'm just scrolling through. Can we comment on Curcumin and bulbar ALS? Yeah. I personally don't see why any product couldn't work for people with either limb onset or bulbar onset disease. You know, we've got ALS reversals that clearly are associated with both types of ALS. And so I wouldn't necessarily reserve a product for one or the other type of ALS unless I had some really good mechanistic reason not to do it. Now, the challenge of bulbar onset ALS is that swallowing becomes difficult. And some of these products, like Curcumin, come in fairly large capsules. Like, sometimes I buy these products myself just to see what they look like. And I might even try them for a few days to see if I have any side effects. And I did that with Curcumin. Capsules were pretty large, but they can be opened and the contents can be put into something that's easier to swallow, or even something that can be put into a feeding tube. One of the problems with Curcumin or any form of curcumin is it's bright orange. If you get that powder on your hands, the fingertips are going to be orange. If you get it on your feeding tube, the feeding tube is going to turn orange. It doesn't mean it's damaged, but some people don't like that color. So let's see if we got any other questions here. Somebody asked about secretions in ALS. It's a great question. So amongst the many things I did not know when I started my career in ALS was how much time I would spend talking about secretions. So it looks like there's two problems that contribute to the secretion issue. One is, it does look like people with ALS might produce more secretions, but clearly they have problems handling secretions. Like, I have become so much more aware of my body since I started taking care of people with ALS. To give you an example, I mean, I've EMG'd myself at least 6 times in the last 21 years because I had a twitch or was wondering if one of my legs was getting weak. I mean, it's hypervigilance, but I've become aware of the number of times that I swallow each day. It's probably several thousand times that I just swallow without even thinking about it because there's saliva in my mouth. And if swallowing becomes more difficult, obviously, now you are aware of the number of times that we have to swallow to clear the saliva that we make. And so I think it's both things. It turns out there's probably two different types of saliva problems in ALS. One is too much of the thin secretions in the front of the mouth that causes drooling. And the treatments that we use for that, well, first we use drying agents. And so those tend to be anticholinergics. And so there's different ones. There's atropine eye drops. The nice thing about those is that they're very short acting. So sometimes patients will have periods during the day when they're too wet and periods when they're too dry. And so I find something short acting is most helpful there. We've got pills like Robinol and amitriptyline that can be used, but some people have difficulty swallowing those pills. We've got a patch called scopolamine that can be stuck on the skin. When those don't work, we've got more aggressive options. We can inject botulinum toxin into the salivary glands. That will drastically diminish the amount of saliva that's coming out for several months. We can even radiate the salivary glands. We can send our patients to radiation oncologists, and they'll radiate the salivary glands. And I think these more aggressive options, which one works best depends on where you are. Like I've certainly had my colleagues tell me at their institution that Botox works great. I've not had as much luck with it at my institution. I've had really good luck with radiation. In fact, the number one side effect that my patients tell me after they get radiated at my institution is that their mouth is now too dry, but they do prefer that to being too wet. The other kind of secretion problem that people get is they get deep, thick secretions in the back of their mouth and they cannot get those up. And so there we use different products. Usually what we do is we start with a mucolytic called Mucinex. It's over the counter, 600 milligrams twice a day. If that doesn't work, we use a nebulizer, and there's different ones. We usually start off with hypertonic saline, concentrated saltwater nebulizer that we can use 2 or 3 times a day. If that doesn't work, then we use acetylcysteine nebulizer. That has to be preceded by albuterol because acetylcysteine by itself can cause bronchospasm. So we got to open up the bronchi before we give acetylcysteine. All that can be put into one nebulizer solution. And then we typically follow any of these treatments with cough assist machines. And so a cough assist machine is something that you put on over your nose and mouth. It's a mask. You flip a switch and you get a machine gun-like burst of positive pressure followed by a vacuum. And that forces you to have a big, deep cough. So if you use some of these other things first, followed by the cough assist machine 2 or 3 times a day, that usually really helps to get up those deep, thick secretions. But very, very common problems, and this is what we do in our clinic. Someone asked for my thoughts on the Radicava infusion. So I think with any treatment, what I try to do for my patients is explain what I see as the potential benefits and the potential burdens. And then I let my patients decide. And if they decide they want it, I try to help them get it. Just want to make sure that they understand what they're getting into. And so, for Riluzole, it's surprising to me how few patients come to me on that. But my explanation is, I think this is the most proven product that we have for slowing ALS progression. Just about every study that's ever been done, and there have been many large, randomized, double-blind, placebo-controlled trials, has shown that people who take Riluzole progress more slowly and live longer compared to people who don't. Now, sometimes patients will say to me, yeah, but I heard it's only another month or 2. Not necessarily. There are some studies that show a 19-month difference between people taking Riluzole and people taking or not taking Riluzole. So we don't know how much an individual patient is going to get out of it, but over and over again, the studies are telling us that people who take it progress more slowly and live longer. The downsides of Riluzole, in my opinion, are very few. It's a pill, it's crushable. There's a liquid form. There's even an oral film if you don't like crushing things. It just has to be taken twice a day. In my experience, most people do not have side effects. Serious side effects are extremely rare. One thing people sometimes worry about is changes in the liver. That's very, very rare, and it's easy to monitor for. We just need to do a blood test monthly for the first 3 months and then every few months after that. And so after hearing that explanation, most of my patients do take Riluzole. Now, when it comes to Radicava, my explanation is a little bit different. There has only been one randomized, double-blind, placebo-controlled trial of Radicava that showed a benefit. This was a study done in Japan, so it probably had mostly Japanese people. The people in this study were in their first 2 years of symptoms. They had a lot of function, at least 2 points on every item on the ALS functional rating scale, and a breathing capacity of at least 80% predicted. And they were progressing at an average to faster than average rate coming into the study. In that study, over the course of 6 months, people on Radicava infusions progressed a little bit slower on the ALS functional rating scale compared to placebo. Now, the only other randomized double-blind placebo-controlled trial I'm aware of Radicava took people who looked a little different, people who looked more like your typical patient in an ALS clinic. So they had the disease a little longer, or they were a little bit worse in terms of their function, or they were progressing a little slower. And that study didn't show a benefit, didn't show any difference compared to placebo. So what I tell people is, I don't know. If you look like the ones in the first study, it's more likely that it's going to help you. But I would have loved to see a third study, because we got one positive study and we got one negative study. And in science, ideally, if you got one positive and one negative, you would do a tiebreaker study. But I don't think that's going to happen now. So I can also say that the burdens of that medicine are much higher than the burdens of Riluzole. It's not a pill, it's IV. It requires so many IV infusions that most people have to have a line implanted in their chest, which is going to cause its own issues. It can cause pain or bleeding or infections or puncture a lung. You'd have to go and get IV infusions for 14 straight days the first month, and 10 days after that every month for the rest of your life. Most of the people in my clinic who've started Radicava have stopped it. So currently about 85 to 90% of the 450 patients in my clinic are taking Riluzole. One of the 450 patients in my clinic is taking Radicava. So that's what I think about those two medicines. Let's see. Another question about AMX 0035, and again, it's unfortunately not available yet. Somebody asked, most of us are taking TUDCA over the counter, but cannot get access to sodium phenylbutyrate. Well, I mean, you can get access to sodium phenylbutyrate. It just requires a prescription from a doctor. The problem is it's expensive. So, I mean, the price ranges anywhere from $1,000 a month to $3,000 a month. The cheapest I've found it is a pharmacy that's in New Jersey, which is called Hopewell. It's a compounding pharmacy, and they're selling it for $1,000 a month. So is it worth it? Well, again, that's a question that I really cannot answer for you. Just like I cannot answer the question, should you take Riluzole? Should you take Radicava? What I can do is explain the data. There's never been a study comparing TUDCA by itself to the combination of TUDCA and sodium phenylbutyrate, not in people anyway. So I don't know if it adds anything to TUDCA. I do know from one study that was done in Europe that TUDCA by itself can slow the progression of ALS. And that's just one study. There's a replication underway right now in Europe, but to me it looks really promising that TUDCA by itself can do something. Whether adding sodium phenylbutyrate adds additional benefit, I don't know. And as far as is that possible additional benefit worth $1,000 a month, I mean, I think that depends on the individual person. Is Riluzole oral film available now? I believe it is. I've never prescribed it to anyone, but I do believe it is. Certainly the liquid, which is called Tiglatec, is available, but I believe the oral film is also available. Let's see if I missed any up here. Oh, tofersen. So someone asked about tofersen. So tofersen is a drug that's being investigated by Biogen. It is something called an antisense oligonucleotide. So it blocks mutant SOD1 genes from producing mutant SOD1 protein. In that form of ALS, which affects about 2 to 3% of everyone with this disease, the production of that mutant protein seems to be the cause of the disease. So this is beautiful in terms of mechanism. This would get an A+ if I was reviewing it from ALS Untangled, because we know exactly what we should do in that form of ALS. We need to knock down as much of this protein as we can. And I'm very hopeful that this is going to have a big effect on the disease. Now, there's only ever been one publication of tofersen in people with ALS, and that was a very small safety study. It was just a 3-month study. It was a handful of patients. But when they focused on the people with the fastest progressing disease in that study, it was kind of amazing. There was no progression in those patients for 3 months. Now, that might not sound like much, just 3 months, but when you looked at the same types of patients with fast progressing SOD1 mutations who were on placebo, their progression rate was like 6 to 8 points per month on the ALS functional rating scale. So 0 progression versus like an 18-point decline over 3 months. That's extraordinary. We've never seen results like that in a clinical trial. So keep my fingers crossed that the next study of tofersen, which is much larger and longer, is going to show us results that exciting. Because if so, it's going to revolutionize the treatment of disease for that form of ALS. Someone asked, is anyone doing neurophoresis in Europe? No, not for ALS. Again, this is pretty new stuff. I don't know of anyone that's doing it for any disease except for the folks at Duke who are studying it for people with subarachnoid hemorrhage. Seeing if I'm missing anything here. I think I might be out of time. It looks like it's 3 o'clock and I don't want to affect the other speakers who are coming after me, but I've really enjoyed spending the last hour and a half with you, and hope that my talk gave you some things to think about. And most importantly, I hope that you walk away with a little more hope maybe than you had before, because I certainly am more excited now than I've ever been in my 21 years. I think we're going to see some much better treatments for ALS in the next few years. So have a good rest of your day. Take care.